my defense is scheduled for next week, so we're crossing our fingers. So I want to tell you today about the work I've been doing using multiple network-constrained regressions to expand our insights into flu vaccine responses. So to begin, the influenza vaccination is widely recommended, but not effective for a substantial proportion of individuals. So in the US, it's recommended for everybody over 6 months of age, but there's a large variability in responses. So what I'm showing in this plot here is the vaccine response measured by the fold change in antibody titers in the circulating blood. And what you can see is that these are young and elderly individuals. Young tend to respond better, but even within each age group, there's a very wide variability in the vaccine response. And because of this, the efficacy is pretty low. It's estimated between 50 and 70% in young adults, and even less than that, the best estimates in older adults are around 50%. And this low efficacy leads to many deaths each year, which are mostly caused by co-infections and pneumonia. So the question we're interested in understanding is what underlying mechanisms characterize a successful vaccine response. And we'd like to know, the simplest way I can frame this is when we give someone the vaccine, we want to know what's going to lead to a high or low response. So in order to answer this question, we've performed a study where we collect blood and collect blood pre-vaccination and then 28 days after vaccination to measure the antibody response. And then we stratify, we take only the extremely high and low responders. And in those individuals, we measure gene expression. This is actually done on microarrays, so this was started in 2010. So in total, we had about 100 subjects to start, and then we took the extremes. And so what I'll show you is data trained on 35 subjects, and then validated in an independent study that was previously published. And I just want to stress that the goal here is not to get the best predictive model. We really want to understand what's leading to vaccine response so that we can design a better vaccine. But one of the major challenges we face is that the high dimensionality leads to multiple equally predictive models. So this is a really nice study here where they were trying to predict symptomatic versus asymptomatic flu infection. And they built the logistic regression model, LASSO, so a sparse model, and they were able to predict vaccine response. And then they took out those genes that were predictive and refit a model again. And they did this for many iterations. And what they're showing is that they can do this for 50 iterations, each time removing 20 to 80 genes before there's a drop in testing accuracy. Okay, maybe this is not surprising to many of you who have worked with this data because gene expression data is highly correlated. The genes are highly correlated. So, but what this tells us is there's multiple equally predictive subsets of genes. So what we wanted to do is use this fact to incorporate prior knowledge encoded in gene networks to improve the interpretability of our classifier. So the way that we did this is to use Laplacian regularization. So the intuition here is that this is our toy gene network, and then genes are connected. For example, if the gene products are in the same pathway. And what we want is that the effect of each gene in the model is smoothed over the network so that they have a similar contribution. But we also might want, for example, genes A and B might have a different direction. So we allow the sign of the coefficient to vary, but the magnitude should be smoothed over the network. And so this is the penalty function where in red I'm highlighting the Laplacian, the network smoothing factor. And it's basically the squared difference in model coefficients. And this was done by Soon and Wang back in 2012. And this type of approach has been used for many years. But we were interested in discovering how this approach would perform on our data using many different types of networks. So we've done this with pathways, ontologies, co-expression networks, functional interaction networks, curated cell subset signatures from the blood, and protein-protein interactions from string. And so we called this method log-minor or logistic multiple network constrained regression. So when we apply this to our flu vaccination study, we find that the log-minor models in the colored box plots accurately predict vaccine response in an independent data set. So it's between 60 and 70% accurate, depending on which model is used. And this is similar to elastic net. We find that the sparse lasso models are not more predictive than random. So the next question is, are the models that use this prior knowledge and networks more interpretable? So we defined the interpretability as the number of enriched gene sets in the model coefficients using a mean rank test. And what we found is that the number of enriched sets is dramatically increased using these prior knowledge networks. So for example, just looking at KEGG pathways, if we use a KEGG prior knowledge network, we get on average 2 KEGG pathways enriched in our predictive models, showing the mean and standard error from 50 iterations of cross-validation. With the standard methods, we get little interpretation in this way, and it's similar to if we permute the labels on the networks, which is shown in the gray boxes. So we did this for multiple types of gene set libraries, and you can see that the log-minor models improve interpretability. So our next question is, is using all of these different networks actually useful? Are they giving us different information? So in order to answer that question, we laid out all of these gene sets that were enriched in our models onto a network. So they're arranged here. So each node in this network is now a gene set, and they're arranged by Jacquard distance on a minimum spanning tree, so that the gene sets that are close in this network have a lot of overlapping genes. For example, each different shape is a different type of gene set. So for example, the cell signatures are circles, and this particular one is B cells. And then we colored each node by which of the models it was enriched in. And finally, we annotated similar groups of gene sets. And if we zoom in on this network, we see 2 things. So one I'll point out is that, for example, the B cell signature is enriched no matter which network we use. Even if we don't use network, the elastic net models also are enriched for B cells, which is not surprising in the context of this problem, because B cells are known to cells that differentiate and produce antibodies that lead to protection against the virus. But we also find some gene sets, for example, interferon signaling, which are only found with a subset of the networks. In this case, Keger reactome pathways or string. Otherwise, we wouldn't find these things enriched. So we conclude that using these multiple models provides context-specific insights. And one of the really interesting applications of this approach is to a drug response network. So we built this drug response network, and the intuition here is the same. We're just going to see if, or we reason that drugs may have an effect on vaccine response if a certain drug signature is enriched in our predictive models. And we were able to identify 6 drugs listed here that we hypothesized might negatively impact a proper vaccine response. So if you look at these, what these drugs inhibit, they're all actually inhibitors of growth factor receptors or members of the RAS-ERK or PI3 kinase mTOR pathways, which are important in growth and proliferation. So we think that this is because growth and proliferation is important for the antibody response. So the inhibitors of these proteins are negatively predicting response. And we actually were able to find in the literature a previous study from 2011, which found that in this scenario where they were treating recipients of solid organ transplants, that mTOR inhibitors did in fact decrease response to influenza vaccination, which is some sort of validation of at least one of our drug targets. So in summary, we propose this log miner method that enables context-specific insights. When we performed classification with standard methods and looked at what was driving prediction, we didn't find many enriched gene sets, which prompted us to develop this method where different sources of prior knowledge are used as sort of lenses to look at the data, and we find that we can develop equally predictive models that are more interpretable and provide context-specific insights. So the code for this is all available on Bitbucket and also linked from the paper. To conclude, I hope I've convinced you that the high-dimensional data, like gene expression data, often contains multiple distinct sets of equally predictive genes, and that log miner is one way that we can use that fact to incorporate prior knowledge and make our models more biologically interpretable, and that we're also able to provide context-specific insights. And I showed you how we were able to identify testable hypotheses about what might lead to high or low vaccine response, for example, that people on mTOR inhibitors would have decreased response to the vaccine. So I'd like to thank all the people who helped make this work possible, the Human Immunology Project Consortium, especially my collaborators at Yale, Albert Shaw and Sabasis Mahanti, my PI, Stephen Kleinstein, all of the study volunteers, as well as funding sources. And if you're interested in learning more, please come visit my poster. Thank you for your attention. Any questions? Yeah. That's right. That's a good question. So the question is, did we use logistic models? And the answer is yes. And the second question is, can we extend this to multinomial models or linear models? And this type of work has been used. For example, the first time in the literature that I know that this approach was used was a linear model. I don't know about multinomial models, but I think it would be possible. Yeah. Yes. The question is, have we looked at multiple strains of influenza? And is there another question?
I can raise my hand.
Okay. Yeah, so I didn't go into the details, but our training data actually came from multiple seasons, multiple vaccination seasons. And as you may know, the vaccine may change from year to year. And each of the vaccines in our seasons had 3 strains. So what we're actually looking for is predictors of response independent of the strain, because there are multiple different strains. That's a good question. Thanks. Oh,
Hi.
I didn't see you, sorry.
I'm hidden behind the pillar, sort of. I had a question about the constraint you're using. So you penalize the magnitude rather than the sign. I was wondering, is this already something that's present in the Lee et al. Paper, or is this something that you've extended yourself?
So this so-called adaptive regularization was presented in this paper by Soon and Wang, and I can give you the reference. So the way that it works in practice is, because obviously penalizing the absolute value of the difference is very challenging computationally. So instead, we estimate the signs of the coefficients beforehand, only in cross-validation only on the piece we're training on. And that's how we're able to allow for a difference in sign. Does that answer your question?
Maybe discuss it for a bit further offline. I have some
Yes.
questions and
Sounds
ideas.
great.
Thank you.
Thanks. Okay, last question.
Same topic, actually. I was just wondering if you had benchmarked it with both restricting the sign to be the same and not, and if you saw any difference.
Good question. As far as accuracy, we didn't see any difference. As far as interpretability, we didn't do a direct comparison. That's a good direction. Thank you.